Okay, hi. Can everybody hear me? Yeah.
Loud and clear.
Hi, Mess.
Hey.
Yeah. Welcome. Good afternoon, good evening, and good morning to everybody who's joining from other parts of the world. Hannah, for example. Yeah, I'm very happy to be giving this workshop today about basic analysis using our custom software BiofilmQ. My name is Eva Jiménez-Siebert, and I'm a PhD student in the lab of Dr. Knut Drescher, Professor Knut Drescher in the Biozentrum of Basel. And I'm going to be talking today about microbial community analysis, basic analysis using BiofilmQ. And I want to point out at this point that I'm not doing this workshop as a standalone. I'm doing it with my two co-hosts who are here today too, Niklas and Hannah Jeckel. Niklas is also a PhD student in our lab, and Hannah Jeckel is a former PhD student from our lab who was part of the original founding team of BiofilmQ, and he's now in the Newman lab in Caltech. So they are going to give two other workshops tomorrow and the day after tomorrow, which I'm going to talk about a bit at the end of this presentation. And you're very warmly welcome, invited to visit those two if you're interested. So yeah, to get started, I need to activate my presentation. Yeah, okay. To get started, I'm going to start with the reason why we developed BiofilmQ in the beginning, in the first place, which is that in our lab, we are interested in looking at biofilms. And biofilms are three-dimensional microbial communities. They are the most abundant form of microbial life on Earth. That alone makes them very interesting, but they are three-dimensional and very tricky to analyze with conventional software. And you can see already here on the right that the heterogeneity of biofilms that you can find is very big. As you can see, the one, can you see my mouse? No, I don't think so. Yeah, okay. Yeah. You can see on the top a large biofilm colony on agar, which can reach sizes for up to millimeters or even centimeters and forms these really beautiful wrinkling patterns. And on the bottom, you can see a very small biofilm, which was grown in this case on a glass surface in a microfluidic chamber and reaches sizes of a few micrometers, and in which we can even see on this rendering the single cells that form part of this biofilm. And the reason we are so interested in these biofilms is that inside of them, spatial gradients can form. And these spatial gradients, they can form in the form of different concentrations of molecules in different parts of the biofilm, but also in the form of spatial gradients of physical properties of the biofilm. And these physical properties can be something like the packing density of the microorganisms that form this community, or the arrangement of these microorganisms to each other. Are they aligned or not? And all of these spatial gradients, they influence the physiology of the single cells inside the biofilm. So this leads to a very high phenotypic heterogeneity in these biofilms, and that is why we're interested in studying them in a more space and time resolved manner. And these two examples on the right don't really cover the heterogeneity that we can see by simple eye when we're looking at different biofilms. As you can see in these different images that were all either acquired or analyzed in our lab, the heterogeneity is really monumental. So we have on the left macrocolonies on agar, macrocolonies of mixed species, but also biofilms inside of hosts, where you can ask the question, how does the biofilm influence the host? How does the host influence the biofilm structure? But also on the more smaller scale, submerged microcolonies or mesocolonies that have on their own very characteristic structure, or pelical biofilms that have already very different rugosity and very different properties that then also influence how the bacteria behave inside of them. And as you can see from this, in our lab, we are interested in analyzing this heterogeneity using tools and developing new tools. And one of the tools that we like to use for that is image analysis. And as you can see from this diversity, it is very difficult to really capture all of this diversity with one single analysis pipeline. And that is why in our lab we use custom software with MATLAB. And we had a lot of different scripts that we kept changing for a little bit for every single new phenomenon that we wanted to analyze. And that is a point where the founding team, or the original creators of BiofilmQ, which you can see here on the right, Aimo Hartmann, Hannah Jeckel, who's here today, and Eric Jelly, came in and thought, can we actually create a tool that is able to capture or to analyze all of this diversity while also keeping it so simple that you can also use it without having any programming skills. And that is why they developed BiofilmQ, which is in principle, in the end, a tool for analysis, image analysis of fluorescent images, and this analysis can be done in a spatially and time-resolved manner. And if you're interested in reading more about it, you can go into the original publication from 2021. And yeah, after having said this, how do you get started with BiofilmQ? So if you Google BiofilmQ, the first three things that come up are these. The first one is the documentation website of BiofilmQ. If you go on there, you can find very useful information about how to use BiofilmQ. You can find documentation on the distinct functions of BiofilmQ. You can also find documentation on how to get started with BiofilmQ. But you can also find tutorials and even video tutorials which guide you through different pipelines, or even with sample data that you can download from the website. You can get a glimpse into how we would use it in our lab. The second hit that you get is the website of our lab, and the third hit is, of course, the paper, and you're very welcome to go into that. If you then click on the first link, this is how it looked a while ago. Now it looks a bit different, but the basic structure should be the same. And I'm going to guide you through the important parts of this website. So one important part is the download tab, in which you can download BiofilmQ and the sample data, of which I was talking about a minute ago. You can download BiofilmQ in different manners. You can either download it as a zip file, as you can see here, or also if you don't have MATLAB, you can download it as an executable file, .exe file, and work with it without having MATLAB. But we recommend, if you want to maybe adapt the BiofilmQ pipelines to your personal problem, if they are not perfect for the application that you want to have, to download the version in which you can work with it in MATLAB. So that is the zip file over here. The second important part is the video tutorials. As I already said, there's a lot of them. You're very welcome to go in there and explore. And the third part that is very important is the forum. We have the image.sc forum, and we regularly look into this forum, and you can go in there if you are having trouble with some of the features of BiofilmQ. If you have any questions, you can go in there and start a thread, or also browse through the threads that are already there and see if anybody has the same kind of question as you. And we regularly look into it and are very happy to answer any questions that come up. And having said that, the basic workflow of BiofilmQ, how the analysis works, is in 3 steps. In the first step, we have a segmentation of the 3-dimensional volume that you input with your raw data. That segmentation is then sliced into cubes and input into an analysis pipeline in which you can calculate different object parameters, in which your objects are the cubes that you sliced in the segmentation. And these parameters that you have calculated can then be visualized with the third section, which is the graphs and visualization section, in which you can then see if maybe the parameters that you calculated don't really give you information about the phenomenon that you want to capture, or even if you have to go a further step back into the segmentation because that one is not really accurate and does not really capture what you wanted it to capture. And yeah, I'm going to go a bit more into this segmentation because that is a very distinct feature of BiofilmQ, and is what is going to enable you to acquire a quasi-cellular resolution, even though you might not have a single cell resolution data. So in the segmentation, there are 3 main steps. And the first step, the raw data that you input is filtered, and you can choose the filters that you want to apply in a customized way. You can choose to use them or not use them. You can choose to change them a bit so they adapt to your data a bit better. And these filters are there to maybe remove background noise or make the signal to noise ratio clearer. After you have filtered, then your data, it gets thresholded, and you can then select or segment your 3D volume, which is this part here. You get a one volume of your biofilm. And after you have thresholded the data, you can slice it into cubes. And this is the distinct part of BiofilmQ. These cubes are also, we call them pseudocells, because depending on the size that you choose for those cubes, you can distinguish different features. And even though you might not have data that has a single cell resolution, you can even get resolution, even get features of the biofilm that might be on a single cell level. As you can see here, for example, by knowing in which position and this cube is and analyzing different parameters for this cube inside the biofilm, we can know how the local density is behaving inside of the biofilm, and that for every part of this 3-dimensional volume. And of course, if you have a good segmentation from a third-party software, or you don't want to filter your data, you can always import data in any one of those steps, for example, from neural networks, and then you can work with single cell segmentation data and use all of the other pipelines of BiofilmQ afterwards. And that is what Niklas is going to talk a bit about tomorrow in this workshop about advanced BiofilmQ analysis pipelines. It's going to be tomorrow at 5 p.m. Central European East summertime. And he's going to talk about single cell segmentation with an example, Stardust OPP as an example for neural network for image segmentation. That this is one that we have also developed in our lab. It's an optimized version of Stardust, but you can use any other segmentation or any other from any other neural network. And he's going to then show you how you can import this segmentation data and do single cell analysis on that segmentation data. This is one of the features that has been added in the new major update that we have done to BiofilmQ in the last 2 weeks. And the third workshop in the series is going to be by Jeckel, and it's going to be about advanced BiofilmQ analysis and visualization. And it's going to be on Thursday, also 5 p.m. Central European summertime. And she's going to talk about the more advanced features of BiofilmQ, like batch analysis tools. For example, if you have a lot of folders on which you want to perform the same task over and over again, you can use the batch analysis tools and have only to click once instead of clicking through every folder. And she's also going to talk about merging, where you can, for example, analyze an image like this one on the right. If you have several channels and you want to do combined analysis of these channels, you can merge them. And she's also going to talk about some of the more advanced features of the visualization that I'm not going to be able to cover today. And then this is the point at which I would go over to the more hands-on part. I forgot to tell you in the beginning, please feel free to interrupt me at any point or ask questions if anything isn't clear. This is supposed to be a hands-on workshop, so yeah. Any questions? I guess not. No, okay. Then I'm gonna share my MATLAB with you. Okay. So you should be able to see my MATLAB window now, right? Yeah, okay, good. So you should all have downloaded BiofilmQ from the website, and you should have gotten a folder which should be called BiofilmQ version 1.0.1. And if you then open MATLAB, or you just click on the executable file, or in this case, like I'm doing it, you open MATLAB, and then you go to this button here, you can then browse to the folder in which you have your BiofilmQ, and yeah, select that folder, and this is how it should look like. And then you can go to the biofilmq.m file and do a right click and run it. And then this is going to start loading now. This is the starting screen that's showing you the status of how much it's loading or not. It's going to take a moment. And you can see here on the command window of MATLAB that it's showing me what BiofilmQ is doing. It's what kind of toolboxes of MATLAB it's using. Yeah. And then now it's done. And you should be able to see my BiofilmQ now. Yeah. Okay. In your case, probably you have a BiofilmQ logo over here and some text over here. I don't have it because I'm using Windows 11, but yeah, that's how it should look like. And now we can go onto this browse button here, and we can choose the folder in which our data is. You should also have downloaded the sample data from the BiofilmQ website, and that should give you a folder which has two things inside. It has a folder which is called sample data and also an ND2 file, .nd2 file. We're going to use the folder for this workshop, but Hannah is going to show you in the next workshop how to import data from proprietary software. So we're going to go to the sample data folder where all of the TIFF files are. I'm not seeing them right now because BiofilmQ doesn't show me that. But then we're going to load the folder. So you can see here on the status bar that BiofilmQ is loading the data inside the folder. And in a minute, it's going to open the main BiofilmQ window. So this is how it looks like. It has two main compartments. It has one compartment for image processing here, the tab image processing, and the second tab is for visualization. After you have done all of your image analysis that you wanted to do, you can visualize it in this tab. But we're going to go back to the image processing tab first, and I'm going to guide you through the different parts that we have in this tab. So on the left, we have three main containers here. We have on the left the files tab, and this tab is showing you the different files that you have in the folder that you browse to. And it's only showing me, in this case, the files for the first channel, because here I have selected the first fluorescence channel. But I can also select the second fluorescence channel, and then it's going to load the second channel. And you can see now here that, oh, yeah, it switched the channel to number 2. I'm going to go back to channel number 1 because that's the one that we're interested in right now. And just to mention it, here you can select also the type of file that you want to see in the folder that you're in. If you select this option on the top, you can import data from proprietary software, for example, NIS or CZI data formats. And then BiofilmQ imports it in a data format that it can work with in TIFF data format. And also what is very important when working with TIFF data is that it has this kind of naming that you can see here. So every file here has a name here in the beginning, which is the same for all of them. Then the channel, which channel it was acquired in, the frame number tells you which time it was acquired on, and then the number of the slices that it has. So BiofilmQ can only work with files if the naming is in this format. And that happens automatically when you choose the import function. If that doesn't work, what always works is to go into the viewer of the software that you're using for imaging and to select the TIFF export and then import the TIFF into BiofilmQ. But then Niklas is going to show you probably tomorrow how to do that. And yeah, so you can see here that when I click through the different files, then in the center tab in the image preview, I can see the image that I'm looking at. And I'm not sure why I cannot click on this right now. But anyways, yeah, this is normally showing you only one channel. And you can click on this display merge view option to then display the one that I'm seeing right now, which is showing both channels at the same time. And you can see when I go through the different files that I'm seeing the biofilm grow. And you can already see here that we have two different biofilms in this image. In the sample data, we have one that has only a red fluorescent protein, and we have another one that has both a red fluorescent protein and a green fluorescent protein. And this is what we're going to analyze today. So yeah, in this image preview folder, another important feature is the show Z-stack feature. When I click on this, I can see the Z-stack for the channel that I have selected and the position or the frame number that I have selected. So you can see here that I can then scroll through the stack with the slider and get a better sense for how the stack looks like. I can also do that again with the last position. And then you can see that I actually can see the three-dimensional structure very nicely.
Eva, can I quickly interrupt you? There's a question in the chat. Did we already go over that?
Oh, sorry. Yeah. Where can we find the folders? What do you mean, the sample data? Okay, the sample data should be on the website.
Yeah, but the link was dead, Eva, actually. It was like 404 not found.
Oh.
But can't you download the sample data in the BiofilmQ download page, where you also download BiofilmQ?
No,
sample
I
data.
don't think so.
Unless it's a different sample data.
Niklas, you're not unmuted. Okay, yeah. So Niklas has just sent a link into the chat, and you can download the sample data from there. I'm sorry about that. Yeah, should I give you a minute or? I'll give you a minute. Are there any other questions until here?
This is for actually opening BiofilmQ, but I'm getting an error saying error using box, too many input arguments. I don't know if that's a common error or that's something strange.
I am not sure about that.
What does the error say? Error using what exactly?
I just sent it in the chat.
Okay.
Oh, I don't think I've seen that yet. So maybe we can talk at the end, or maybe we can like figure it out aside. And if I can move on in the meantime.
Yeah, it might really be about the MATLAB version. BiofilmQ was written in 2017, so.
I'm using 2023.
Okay.
And it's working, so I'm guessing there might be something else, but I would suggest that
yeah,
we think about it or look at it later.
Yeah. Okay, so now you should have downloaded the sample data, or if not, I'm gonna move on, I'm sorry. But yeah, anyways. So you have the preview here, the image preview, and also an important thing to notice is here, the scaling. It should be recognized automatically by BiofilmQ, but if it isn't, for some of the calculations, it's important. And yeah, if it is not recognized automatically, you can change it here. That is the pixel size in nanometers and the distance between your Z slices. And then you should click update all images to apply the settings to all images. And yeah, I'm gonna go into the other features later when we have actually calculated something. The section on the right is the one where the main part of the image analysis is gonna take place, where all the calculations are gonna take place. And also here you can see different tabs. They are ordered in such a way that they follow the logical process that you would do while segmenting the images. But you can, of course, if you don't need one of them, skip them and just go to the next one. We have the image preparation, in which we're going to do some pre-processing of the data before the segmentation, the actual segmentation. We have parameter calculation afterwards, in which we can calculate parameters for the segmentation that we did. We have a time series analysis. I would recommend to use this one for cube tracking. There's a tutorial for that on the website, but not for more elaborate single cell tracking. For that, you can use third-party software like TrackMate, for example, that is very good at that kind of thing. And you have a data export in the end where you can export the data that you calculated as for 3D visualization, for example, with ParaView as VTK files, or as flow cytometry files, or also as Excel and CSV files, in case you are more comfortable working with Excel. So I'm going to go through them step by step and explain the most important features that we can find in these tabs. In the first tab, you should be especially interested in the image analysis alignment. So you can see when I click through the different positions here that between the different positions, the image moves a bit, and that is quite normal and usual while doing microscopy, that you have some kind of drift between the different positions, but you can correct for that if you want to do some analysis that requires you to. And for that, you need this image alignment. And one important thing to see, so this image alignment takes one image, a reference frame, and then tries to align all of the others to this reference frame. And that is what you can choose in this window here. So it's recommended to take as a reference frame the frame in which the largest object is that you want to still be able to analyze. And in this case, it's the last frame. So it's the frame number 7. And that is why in this case, the reference frame is frame number 7, but usually the default here is number 1. And then you could, if you don't want number 1, but you want number 7, you could input here 7 and click on set current frame. And here you can choose which kind of alignment you want to do, which kind of registration method. Correlation is the most usual one. And you can choose also to align it along Z in case you have some drift also along the Z position of your images. And then you can click on register images. That is going to now open a window that is for me in the other screen. You can see now the window in which BiofilmQ is showing you the translation, the movement between the different positions that it is correcting for. So position number 1 was here, position number 2 is here, 3, 4. And you can also see here what BiofilmQ is actually doing. It's aligning the images according to a reference frame in X, Y, and also in Z, which you're seeing right now. And now it's done with the image registration. And we can now click through the different images and see that now there is no movement anymore. So the image registration has worked very nicely. Okay, so once this is done, you can go over to the segmentation part. And here is where your objects are going to be actually segmented. We have 2 different parts, 2 different types of segmentation that we can do here. We have thresholding segmentation. That is the standard BiofilmQ segmentation that I was talking about earlier in the presentation, but we also have label image segmentation, and that is the part that Niklas is going to cover tomorrow, where you can import segmentation from other software. So I'm not going to talk about that one. And then here we have the different steps that you can do for the segmentation. You have pre-processing, so you can denoise, you can correct for certain things that your microscopy setup might have. You have the thresholding, the declumping, where you can select the cube size, where you can basically dissect your volume into the cubes, and you have the post-processing, in which you can again correct after having segmented for issues that you might have. So yeah, cropping is the first thing that you can do. This is very useful, especially, oh yeah, one thing, you can select here if you want to apply the image registration or not that you calculated in the first step. So if you check this box, then the image registration is going to be applied for your calculations from there. This is one feature on this tab, and the main feature on this tab is the cropping. So cropping is very useful doing image analysis in case you have, for example, very big images, or in case, and you're only interested in a subset of the image, or in case you have some background cells like here, for example, which you don't really want to account for in your analysis. And you can choose a cropping for all of the images in the folder that you're working in by either putting in the numbers here that you're going to see in a minute, or what is much easier, you can click on select, and then you get a window which opens that shows you the image that you are currently selecting, and that gives you the option of making a cropping. Also here, you should crop it to the size of the biggest structure that you want to analyze, of course, in the course of your time lapse. So that would be, again, the last frame here. And if I repeat the selection with the last frame, I can then select the part of the image that I'm interested in, and that is going to only analyze that part and make the analysis much faster. So let's say, for the sake of the argument, in this case, I'm interested in knowing how does the biofilm that has both fluorophores, the GFP and also the red fluorescent protein, develop over time. So I would draw a rectangle to crop that biofilm. And then that cropping would be applied. You can see it here. And also these are the numbers that I was talking about earlier that you can input into this. These numbers tell you the bottom corner of this cropping rectangle, the width and the height of the cropping rectangle, but it's much easier to just select it. As you can see, when I click then through the other files, they don't have the same cropping. You can select different croppings for the different files, but you can also just click apply to all images if you want to use the same cropping for all images, and then you can just go through them and see that the cropping was applied to all of them. But a nice feature that BiofilmQ has that is very useful in this case is that you can also dynamically define your cropping by just defining a beginning cropping and an ending cropping, and then you can tell it to interpolate between those two beginning and end croppings, and it will automatically define the cropping itself. So for that, I need to renew the cropping. That's why I'm deleting this and pressing enter. And I'm going to click again, apply to all images. So now they have no cropping anymore. And then I'm going to go again to the last image. I'm going to crop the biofilm that I'm interested in. I'm not going to click apply to all images. I'm going to go to the first one. I'm going to select here also cropping. That's going to be the size of the biofilm in the beginning. And then I'm going to click interpolate crop rectangles. And when you do that now, through clicking through the images, you can see that the cropping rectangle is adapting to the size because it interpolated between the two ones that I gave it. So this is not only going to save us computation time, but it's also going to get rid in the beginning of these cells that I'm not really interested in and just analyze the one that I'm interested in. You can also tell it to do a Z cropping, but we're not going to do that today because our files are not really that high. They are easy to process, and in case you might be interested. And you can also adapt the direction of the flow, which right now is in that direction. Then the next step that you can do is some corrections for your optical setup. For example, invert stack along Z, that would be the case. BiofilmQ assumes that your biofilms are growing from the top to the bottom, and it always analyzes them like that. But there are also setups in which the biofilms grow from the top. No, it assumes that they grow from the bottom to the top. Sorry, I said it wrong. There are also setups where they grow the other way around, and by clicking this, you can correct for that, and BiofilmQ is then going to be able to work with them from there. You can also correct for a tilted cover slide, or you can also do some up or down scaling in case your image is very large. You can downscale it by choosing here a value lower than 1, and then the computation time is going to go down. But you can also choose to upscale it by choosing a value higher than 1, and then additional pixels are going to be added between the ones that your data has. So this is not going to add actual information. It's not going to increase your resolution. It's just going to give a finer grid on which to segment. We're not going to do any of these because we don't need it for our case. And now we're coming to the filtering, to the denoising. And here we have 3 different filters that we can choose. We have the first filter is a simple blurring filter, which just blurs the image, and this very effectively gets rid of salt and pepper noise, which is small variations in pixel intensity. And you can choose the size of your blurring filter here. You have 2 numbers. The first one is the size of your blurring filter in X and Y, and the second one is the size of your blurring filter along the Z-axis. You can also check for demand here, but what we recommend to do more is to get a visual feedback for how these filters look like and if they give you the kind of analysis that you want to have. And that is something that we're going to be able to do in the next step. So I'm going to show you in a minute. In general, while doing image analysis, it's always very, very important to get a visual feedback on what you're doing, because your algorithm is most likely always going to segment something, but it's not going to tell you, hey, I'm segmenting something that I should not be segmenting. It's just going to give you some number. And if you don't actually check that it's doing the right thing, then you might get some gibberish. So the second filter that you can use here is a medium filter along Z. With this medium filter, you can get rid of floating cells. So this just checks if a cell is only present in one slice of your Z stack, then it's most likely a cell that is not part of the biofilm and that is just swimming around or floating around, and you can get rid of them by using this filter. And we're actually going to use it for this case. The standard is that these 2 are checked. And the third filter is most recommended for cases in which you want to do single cell segmentation. Sorry, I was looking at the chat. The top hat filter is used for cases in which you want to have single cell segmentation. It gets rid of noise in between cells very effectively. And in this case, I'm going to leave it there because in this case, it actually makes sense. We're not going to do a single cell segmentation, but it's going to get rid of noise in our case. And you can actually get a visual feedback on what you're doing with these filters on the next tab, on the thresholding tab. You have here, you can select different methods of thresholding your images. You can select some of the automatic methods where the threshold is detected automatically and then your data gets put into background and foreground automatically. Or you can select the manual threshold, in which you can just select some threshold and then that's the one that is used. This is an intensity value, so every intensity above this intensity value is then detected as a cell, and everything below this intensity value is background. And you can get a feedback, a visual feedback on how this looks by clicking on this button. So when you click on this button, you should get a preview of what you are doing with the filters that you're applying and with the threshold that you're applying. And in this case, you can choose 2 different options here. You can choose to get the preview without the filters that you're applying in the previous section here, or with the filters that you're applying. What I just click is with the filters, and that is right now awakening my parallel pool. So that's why it's taking a moment. Yeah, but now you can see that it opened a window in which I can see the part that I cropped out of my biofilm, and I see a blue background. Maybe just a quick introduction into this kind of view. You can see in the center the XY slice that I'm looking at right now.
Eva, at least I'm not seeing anything. The window
You're seeing it?
No,
Okay.
I don't know if I'm the only one, but
Yeah. No. Thanks. Now.
yes.
Yeah.
Thanks.
Okay. Sorry about that. Then you probably also didn't see the Z stack, right? Never mind. Okay, so again, you see in the center, this is one of the slices of my three-dimensional stack. And on the side, you can see the side view while slicing the stack through this line, and the same thing here in the horizontal way. You can see the side view of the stack when you slice through this point here, through this line. And I can actually click through this XY projection, and I can get different slicings. But I can also scroll with my mouse through the stack, and then I can see the stack itself. And what you're seeing here is a blue background and some cells. So the background is what is being detected by my thresholding algorithm as a background. And the parts that are gray or whitish or a bit black, those are the parts that my algorithm is detecting as foreground and as a volume that it wants to segment. And I can actually here, by using the slider, select a different threshold. So I can say, okay, this looks like it's actually like it's segmenting not enough of the volume, and then go down and select the one that looks best to me. So in this case, I would select it like this. And as I said, this one was after applying the image processing, but you can also choose to not apply it. And probably you can also not see this one now, right? No? Good.
No, we don't see any.
I think I'm actually, gotta do it like this. Now you should see it. Okay. Now you can see that I didn't apply any filters, and this is much, much noisier, but it's much faster, because before opening this window, MATLAB didn't do anything but just loading the image and previewing it. But you can also see here that I can also choose my threshold, but this is not reflecting what the whole segmentation pipeline is going to do when I actually go click on segment cells and it applies all of the filters. So if you can, it's recommended to use the second option. And now this is the point where we can see what effect these filters have on our segmentation. So we can, for example, choose to make this filter here bigger. We can do the XY filter, for example, 13 pixels, and we can go here. We can click this again, and then we can see that the image looks much blurrier now, even so blurry that it's too blurry for what we want to have. But you can then also go back and put the filter size back to how it was. And a nice thing about BiofilmQ is that now you can click on this again, and you can actually get both of the previews side by side. And these are synchronized with each other. So when I scroll through the stack here, I can see both of them. And you can see that the one where I applied the bigger filter, the bigger blurring filter, is much more blurry than the one on the right. And this doesn't really reflect the data, but it might be useful in some cases. Like, for example, if you want to just have a rough outline of your biofilm, you might want to apply a bit of a rougher filter, of a bigger filter, than if you want to have a single cell segmentation. Okay, there's a question in the chat. Are the thresholds independent of the fluorescence channels? Or are we currently looking at channel 1? We're currently looking at channel 1, yes. If you wanted to segment channel 2, you would have to click here on channel 2 and then do all of this again. So yeah, because it would make no sense to segment 2 channels with the same threshold if maybe the first channel is a constitutive channel with a very strong signal, and the second channel is maybe a reporter that sometimes is not even on, and then when it's on, it has a lower fluorescence than your constitutive signal, or even a higher one. So that's why, yes, you do need to have to do all of this independent from each other for every channel. But you can also do analysis of the fluorescence on the segmentation of the first channel. I'm not going to go into that today because we don't really have time, but that's a possibility if you're interested in that. Yeah, we can talk about it. I'm going to close this now because I'm seeing we only have 15 minutes left. But that is how you could get a visual feedback on what you're doing in this section. And in this case, I want to actually apply an automatic method because I don't want to select the threshold manually for every image, and that you can do with these other methods. The most used one is OTSU. And for the OTSU method, you can choose either to have two intensities, so to have one threshold and 2 categories, 1 foreground and 1 background, or to have 3 categories, 1 foreground and 2 background. And you can, of course, choose which one, if the middle category is going to be foreground or background. But we're going to use 2 for this case. And one important parameter that you can set here is the sensitivity. This should be 1 for you, I think, the standard. You can also change this by, again, opening this window. And the sensitivity, what it's going to do is, if by applying the automatic thresholding with OTSU, you always get a threshold that is a bit too high or a bit too low, you can change it with the sensitivity so that it gets a bit lower or a bit higher. And with the standard parameter, that would be 1, it would look like this. That would mean that my segmentation, my automatic segmentation would be too much segmentation. So I would get a very low volume segmented, and I would have to turn the sensitivity down until it looks the way I wanted. And in this case, I would go for something like this. And then this is going to select the threshold for every image automatically, so I don't have to do that anymore. So moving on from this to the next step, this is where you can, after your 3D volume has been segmented, declump it into single objects. This is where you can get your spatial resolution into the parameters that you want to calculate. And you have 2 options here again. You have the option to use cubes for declumping, which is the standard way, or you have also the option to not use any declumping. This can be useful if you are interested in the whole volume as one and you don't actually want any spatial information, or if your segmentation is already very, very good by just applying the filters and thresholding, and you get a very good separation of the single cells, then it can be useful to not do any declumping. But in this case, we're going to go for the declumping. And here it's important how you choose your cube size length. So your cube is what is going to give you, as I said, your spatial resolution, right? So that means that you should choose the size of your cube to be a bit smaller, or so the rule of thumb is to choose it as large as possible, because the smaller your cubes are, the more cubes you're going to have and the more computational power you're going to need for all of the steps afterwards, and also for the segmentation. But it should also always be a bit smaller than the lowest feature, the smallest feature that you want to actually resolve. So if I want to look at a feature that I expect to vary on a cell level, then I should choose the cube size to be a bit smaller than the size of my cells. And that is what I'm going to do here. I'm going to choose a cube size. So this is the length of the cube. I'm going to choose a cube size that is roughly 1 micrometer. This is 16 voxels, and you can see the size, the cube side length here in micrometers. This is, for example, one thing for which the scaling here is important. And yeah. Okay. The last tabs give you some post-processing options. You can either use a 3D median of the segmented image that is kind of the same as we had here in the denoising tab, but on the binary image after having segmented it, after having thresholded it. We're not going to use that. But an option that is always very useful is to remove small voxel clusters, and you can choose the size. You can basically choose here a size below which you can remove after segmenting every little object in your segmentation. And you choose this size by knowing kind of how big is the smallest object that you're gonna segment. And then you can, for example, here know that everything that is smaller than a cell is most probably going to be noise. So you can choose the amount of voxels here to be, for example, here 0.1 cubic micrometer, and you can get rid of all that noise. And this option is one that you can use when you, for example, know that your image quality is going to be bad after having a certain amount of objects in your image. Like, for example, here, 6,000 cells or objects, cubes. You can select this, and then the segmentation will stop after this frame with the assumption that all the frames afterwards are going to have a bad image quality. So you can choose this option if you want to save time while doing the computation. But in this case, we don't really need that because this is going to be very fast. So we can also remove slices. I'm not going to go into that. And the last tab is about merging different channels. This is a feature that Hannah is going to tackle in her part of the workshop, because this is one of the advanced features that is going to take a bit of time to explain. And after going through all of these tabs, you can then click on segment cells. And before you do that, it's important to look at this here. This gives you the range of images on which, in general, any operation in BiofilmQ is going to be made. So in this case, 1 to 7 is selected. That is all images. But you can also say that I only want to segment, for example, the first image. Then I would remove this and just leave the one. Or if I wanted to segment several images that are not successive to each other, like, for example, 1, 3, 5, you could do that by just inputting the numbers with a space in between. You can also do this by just selecting the images that you want here and clicking here on selected images. In this case, I want to analyze all of the images, so I'm going to click on all images, and I'm going to click then on segment cells. And this is going to take a moment. So while the processing is happening, you can see in the MATLAB command window what BiofilmQ is doing. While the processing is happening, I'm going to go over to the parameter calculation and explain it a bit to you. So here you can calculate on the objects that you have segmented in your previous tab, on the segmentation tab. You can calculate different object parameters. And there is two main classes of parameters that you can calculate here. There is global biofilm properties. Those are these, the number 7. And the global biofilm properties are calculated for the whole biofilm that you're segmenting. So this is without the cube dissection. It just gives you parameters like how much biovolume is there in total, or what is the height of the biofilm, or what is the surface area of the biofilm, that kind of thing. The rest of the parameters are calculated on a cube level. So that means that for every single cube, it calculates, for example, what is the density around this cube, or what is the fluorescence of a certain channel in this cube, in the segmentation of this cube. I'm not gonna explain all of the single parameters because we don't have the time for that today. But if you click on them, you can here in the bottom. Okay, right now it's not doing it. If you click on it, you can see here a description, a short description of what the parameter does, what it's calculating. And also here you can input different options for the different parameters. For example, you could calculate the distance of all of your cubes to the nearest neighbor in a certain channel, and then you could select which channel you want to calculate it to. In my case, I want to calculate the global biofilm properties, and I also want to calculate the surface properties, and I also want to calculate the distance to the center of the biofilm. And this is going to happen in 5 seconds when it's done. Are there any questions to that part until now?
Maybe I missed it, Eva, but
Yeah.
is there a rule of thumb in terms of when to apply OTSO or manual or any of the other thresholding segmentation methods?
Yeah. Yeah, that's so if your image quality is very similar between your different time steps, then you can very well use an automatic option. Because what it does is basically it detects based on the distribution of the intensity values in your images where the background begins and where the foreground begins. If the image quality, if the fluorescence quality is very different between the different time points, you can still try to do that, but it might get to a point if you have a lot of bleaching, for example, where you might have to use a manual threshold. I think that is kind of the rule of thumb. I don't know if Anna or Niklas have other opinions on that, or no, doesn't seem to be. So yeah, if you have very different fluorescence intensities, not really intensities, but distributions in your data between the different frames that you want to analyze, then you should use a manual one.
But would you then do a manual for each time step?
Yeah.
Okay.
Maybe to add.
It always depends on the accuracy that you want to have, right? If you just want to coarse segmentation of your biovolume and just want to know coarsely how much is there, then it might be fine to do an automatic one.
Do you hear me?
Yeah.
It also might really depend on the different image intensities or the intensities. If you have cells that are green, but some that are very green and some that are less green, and then there's the background green, then this might be problematic. But if you have like a very unimodal distribution of the bright cells that you want to segment, also I think it should work. But if you have a very wide distribution of the bright cells, then you have to play around with this adds on this.
Okay.
There's also a question in the chat. So the question is, how different is BiofilmQ from ComStat? I cannot really tell you that because I don't use ComStat myself. How can we segment? Yeah.
Or maybe I can answer to this very quickly. So it is different from ComStat in mainly that it has the wide visualization part. Also, the spatial context is something that you can't really have in ComStat. We do actually have included all of the measurements that you can do in ComStat also in BiofilmQ, and there is a table on the website in the documentation on how to get these properties. So in principle, you would be able to do the same type of analysis in BiofilmQ that you can also do with ComStat, and then there's some stuff in addition.
Thank you. The second question is, how can we segment individual bacteria in packed biofilms? yeah, that is exactly the problem, right? There's a lot of development on imaging techniques to be able to do this. So this depends very strongly on how good the intensity to noise ratio of your data is. And yeah, you can try to counteract a bad signal to noise ratio with image analysis, but there is definitely a limit. And yeah, the most important thing is basically how good is your data. You can, of course, do some deconvolution. There's software for that, for example, Huygens, where you can counteract for imaging artifacts of your microscope. And there's also pipelines with neural networks where you can try to get rid of a noise that might prevent you from doing a single cell segmentation, a good one, with care, for example. But yeah, there's definitely a limit. It depends strongly on your data. And what are the chances of recording artifacts at single bacterial level segmentation in dense biofilms? Depends again on your data and your segmentation. For single cell segmentation, I would recommend to go to the workshop tomorrow with Niklas. Because yeah, some of the neural network algorithms are able to handle even quite packed images by basically giving them a ground truth and learning probably factors that you cannot capture with a classical image analysis. Like all that I've been talking about today is a classical image analysis. So it's based on deterministic algorithms that you can use, filters that you can put over your images. But yeah, some of the neural network softwares might be able to segment single cell, packed single cell communities better. Yeah, I think that's it. There was something else. Yeah, again, get a visual feedback on if your segmentation is good or not. I didn't tell you about this, but you can go here, for example, on this button overlay, and this is probably going to take a while to load, but this is going to show you your data overlaid with your segmentation. And by looking at this, you can see if your segmentation is good or not. So here you can also see that the cells have been neatly gridded into these cubes. So yeah, get a visual feedback on if your segmentation is good or not. And then from there, you can move on. If your segmentation is not good, the chances of getting artifacts are high. But that is also one of the good things about using cubes, that you don't really care about a single cell segmentation. You just care about, is there biomass or is there no biomass? And then can I calculate features for the biomass that there is? And detecting biomass is much easier than actually doing a single cell segmentation. And another question, can we integrate segmentation tools like cell pose environments in BiofilmQ? You cannot directly integrate them, but you can use cell pose and then import the segmentation from cell pose into BiofilmQ. That is, again, what Niklas is going to talk about tomorrow. So looks like his workshop might be very useful for you. Yeah, sure. You're welcome. Okay. So I'm going to calculate these three properties. I think, yeah, I'm over my time. I'm sorry for that. If it's okay for, you're free to stay. I'm going to talk a bit more about how you could continue with this kind of thing, but it's not going to take long. So yeah, I wanted to calculate these. I think I didn't. No, I didn't. So I'm going to click on calculate the surface properties. Yeah, you can, this is going to give me things like the biofilm thickness or the roughness of the biofilm outline, the global properties, and the distance to the center in the biofilm. And I'm going to show you how to visualize those properties and a quick introduction into the visualization tab, and that's going to be it for today. So I'm calculating these different properties now. I'm going to take a moment. And then maybe I can give you already an introduction into the visualization tab. So here you have again on the left the different files that you have in the folder that you're using. And you can click here on load data. That is going to load the segmentation data that you have calculated in the first step, not the raw image data, but the segmentation data. You can limit the frames you want to import to a maximum frame number if you want to, again, save computational time. And then here, you're going to get a list of the parameters that were calculated for the data that you imported. And you can select these parameters and different types of graphs that you want to do with them here to then plot them. And it looks like the parameter calculation is done now. So a nice feature that you can use with BiofilmQ is to go here into load segmentation results. And then you can see the parameters that you have calculated, and you can actually get the ortho view, the Z-stack with the parameters as a color coded. So one that is always very nice to look at is the distance to the biofilm center, for example. And you get, again, this window through which you can scroll through. And you can see when I go to the bottom that in the center, of course, there is nothing because the distance to the center is 0 in the center. And the further away you go, the more the color goes toward this yellow, which if you look at the color bar means that the distance is higher. So this is a very nice feature in which you can also get a visual feedback on if your calculation of parameters makes sense. And after you have done all of this calculation, you can go into the visualization tab. And here you can load the data that you have calculated. And in this case, I'm going to do a very simple plotting of a 3D heat map, a chymograph. So as I said, on the left, you can see the different files. I can also here select the image range on which I want to plot, but on this case, I want to plot all of them. And then I can select here what I want to have in the x-axis of my plot, the y-axis, and then color-coded another parameter. I have 3 options here because I selected here that I want to have a heat map, but I can also tell it that I want to do a 2D plot, and then I only can select 2 parameters that I want to have on the x and the y-axis. I'm going to go back to the 3D heat map, and I'm going to choose on the x-axis the time, on the y-axis the distance to the biofilm center at the substrate, and color-coded, I'm going to choose the local thickness. This is how high the biofilm is, and I'm going to choose the mean here. So here you can also select if you want to have the mean or the standard deviation or the median of the data that you're plotting of the cubes that are being binned together. And then you can click here on plot, and you get this nice chymograph, and you can see that as expected, in the x-axis you have the time, in the y-axis you have the distance from the biofilm center. As expected, the maximal distance to the biofilm center along time increases because the biofilm outline is growing. And you can see that in the center of the biofilm, especially at later time points, the thickness of the biofilm is higher, which is also what we were expecting. And on the outside, the thickness is lower. So yeah, this is a simple example of the kind of plots that you can do with BiofilmQ after you have done all of the image processing. But I hope it showed you that it's a very powerful tool to get a visual feedback, a very fast visual feedback on if what you are doing makes sense or it doesn't. And I think this is as far as I'm going to go today, because I'm already 10 minutes past the time. Are there any questions? Okay. If not, then thank you very much for listening and for staying with me so long, even over the time. And yeah, I hope it was useful to you. I hope you have fun using BiofilmQ, and definitely reach out if you have any questions. We're always very happy to help.